Deep in the forest, a carpenter ant abandons its colony, descends the tree trunk, and climbs onto a specific plant stalk. It stops exactly 26 centimeters above the soil, finds a leaf, and clamps its jaws onto a major vein with such immense force that it becomes permanently anchored in place. Shortly after, the ant dies. We know this highly specific, eerie sequence of events has been happening for a very long time. Paleontologists searching the Messel pit in Germany found fossilized leaves bearing the exact same dumbbell-shaped bite marks. Those fossils are 48 million years old. The culprit behind this behavior isn't a virus or a genetic defect. It is a parasitic fungus called Ophiocordyceps unilateralis. The fungus forces the ant into this exact position for a reason. By commandeering the insect's body, the parasite guarantees its own reproduction, turning a living host into an elevated launch pad to scatter its spores. To find this microscopic puppeteer today, you have to look in warm, highly humid environments, primarily the dense tropical rainforests of Brazil, Thailand, and Australia. The British naturalist Alfred Russel Wallace first discovered the fungus during his expeditions in 1859, observing its bizarre fruiting bodies sprouting from dead insects. For decades, scientists treated the zombie ant fungus as a single species, but modern morphological and genetic research reveals it is actually a species complex. This means it is a collection of distinct, highly specialized fungal subspecies, broken down into different clades, or genetic branches. These branches operate very differently. The O. unilateralis core clade, for instance, specifically targets carpenter ants and forces them to bite onto leaves. Meanwhile, the Nymphophioides subclade targets neotropical ants, which don't exhibit the biting behavior and instead die among the moss carpets on the forest floor. This extreme biological targeting shows us that the fungus has evolved at the subspecies level. It doesn't use a one-size-fits-all attack. Instead, it perfectly exploits the unique behaviors and biology of one specific host ant to ensure its own survival. The infection timeline begins when an ant accidentally brushes against a fungal spore. The spore attaches to the insect's hard outer exoskeleton and immediately begins to drill. Using a combination of sheer physical pressure and specific enzymes that dissolve proteins and fats, the spore breaks through the armor and infiltrates the bloodstream. Once inside, the fungus converts into a yeast stage and begins to multiply. These cells circulate through the ant's body, strategically gathering in massive populations directly alongside the host's brain. Here, the fungal cells begin their chemical takeover. They secrete highly specific chemical compounds, or metabolites, such as sphingosine and GBA, that interact directly with the ant's central nervous system. The physical symptoms of this chemical flood are violent. The infected ant begins to suffer irregularly timed full-body convulsions. These spasms physically dislodge the insect, forcing it to fall out of its safe, elevated canopy nest and down to the dangerous forest floor below. The fungus never actually consumes the ant's brain. Instead, it chemically pilots it. The parasite keeps the host alive and aware, turning the ant's nervous system into a biological vehicle steered entirely by fungal chemicals. Once the ant is steered to the correct leaf, the fungus executes the famous death grip. Fungal cells physically infiltrate the ant's jaw muscles, severing the microscopic connections between the muscle fibers. At the same time, the fungus drains the host's cells of mitochondria, entirely cutting off the jaw's energy supply. The result is total and permanent muscle atrophy. The ant is entirely unable to open its mandibles, locking it in place upside down on the leaf vein as it finally dies. With the host dead, the parasite begins a phase known as somatic investment. To protect its new incubator from scavengers and decay, the fungus secretes potent antimicrobial chemicals and structurally reinforces the ant's exoskeleton from the inside out. 4 to 10 days later, the reproduction cycle peaks. A large spore-bearing stalk bursts from the back of the ant's head. When it matures, it finally ruptures, showering fresh spores onto the dense graveyards of previously infected ants below. That highly specific 26-centimeter height the ant was forced to climb is no accident. It provides the exact temperature, humidity, and elevation the fungus requires for maximum spore dispersal. Yet the ants are not passive victims. Over millions of years, they have developed their own countermeasures. Colonies practice social immunity, actively grooming each other's exoskeletons to remove spores. If an ant senses a nest mate is infected, healthy workers will physically carry the diseased ant far away from the colony to contain the spread. They also use spatial defense. Carpenter ants build extensive aerial trail networks high in the canopy. They specifically construct these paths to avoid walking on the forest floor, entirely bypassing the fungal graveyards waiting below. But the most effective defense mechanism comes from the ecosystem itself. The terrifying zombie fungus suffers from its own parasite. It is routinely attacked by an unidentified hyperparasite, an anti-zombie fungus. As the zombie fungus attempts to grow its reproductive stalk out of the ant's body, the hyperparasite moves in, chemically attacking the stalk and preventing it from ever releasing its spores. This data visualization shows the massive statistical impact of the hyperparasite. Out of an entire population of zombie ant fruiting bodies, only about 6.5% actually survive to produce viable spores. Between the ants' social grooming, their elevated trails, and the aggressive hyperparasite, the ecosystem severely limits the reach of the zombie fungus. It prevents the parasite from wiping out entire colonies, ensuring the jungle remains in equilibrium. The very same chemical weapons the fungus uses to break insect defenses are now being studied in human laboratories. Researchers are specifically looking at the secondary metabolites, the organic compounds the fungus secretes to manipulate its host. By isolating compounds like naphthoquinones and polyketides, scientists are discovering powerful bioactive properties. In clinical trials, these chemical derivatives are showing active results against malaria. They are also being rigorously tested for their antibacterial properties, as well as their ability to inhibit tumor growth. The bright red naphthoquinone pigments produced by the fungus are exceptionally stable under acidic conditions and are completely non-toxic to humans. Because of this, they are being studied as a supplemental treatment for secondary tuberculosis patients to help enhance immunity and improve symptoms when combined with traditional chemotherapy drugs. By regulating insect populations and supporting the survival of other fungi, Ophiocordyceps unilateralis acts as a stabilizer for the rainforest ecosystem. Its specialized chemical toolkit is now being adapted for pharmaceutical research, providing a foundation for experimental human medical trials.